Hello, my name is Giuseppe Trigiante, and I work in the Center for Cell Biology and Cutaneous Research in Queen Mary University. I am here to illustrate a work we've done together with my collaborator Trigiante and Professor Mike Philpott regarding the interfollicular penetration of hydrophilic compounds. Now, the main reason for this work is that many diseases, skin diseases, are connected to the hair follicle, from excessive hair growth to alopecia, and it would be desirable to deliver molecules, especially hydrophilic molecules, which are the majority of interesting drugs, into the hair follicle, while at the same time sparing the surrounding interfollicular epidermis. Now, the hair follicle is difficult to reach, first because it's embedded deep in the dermis, and because the entrance to the hair follicle is normally occupied by the keratinized hair shaft, which prevents penetration of most compounds. The way to deliver things to the hair follicle, the follicular delivery, is therefore a complex problem. We decided to tackle this problem by exploiting the fact that when the hair shaft is lost by a process called epilation, the follicle itself remains empty or vacant. This happens every time an epilation is carried out, or naturally at the end of each hair growth cycle phase. Now, as you can see in this diagram, the hair follicle removal, the shaft removal, results in an empty hair follicle. However, unlike what the scheme suggests, the infundibulum, which is the canal leading to the hair bulb, doesn't remain completely empty, but it's actually clogged with material deriving either from the interfollicular epidermis or from the sebaceous gland. This material is mostly hydrophobic in nature and prevents the penetration of any water-based solution. On top of that, the entrance to the hair follicle is very narrow, and this fact alone prevents the penetration of watery solutions, which are not able, because of surface tension, to adapt to such a narrow passage. So what we've done is develop a formulation based on organic material, such as short-chain esters and alcohol, which on one hand has a low surface tension and can therefore adapt to the infundibulum of the hair follicle, and at the same time is able to penetrate the hydrophobic material at the entrance of the hair follicle and all the way down to the root. This formulation is designed in such a way that it can actually be mixed with a hydrophilic, a watery solution containing active principles, and make the whole formula hydrophobic enough to penetrate the hair follicle. Another twist to the idea is to apply the solution to the skin prior to epilation, prior to the removal of the hair shaft. So as you can see here, this process, the removal of the shaft, creates a depression within the hair follicle. If a solution is present outside the skin at the moment of removal of the shaft, this solution would be pulled into the hair follicle and reach the bulb, where it can actually carry out its activity. The way to test this idea is to carry it out on ex vivo explanted skin. We use facelift skin, and treat it with a formula containing a test active whose penetration in the hair follicle could be easily measured. What we decided to use is hydrogen peroxide, which is a very simple molecule, very highly hydrophilic, and whose presence in the hair follicle would result in damage to the hair follicle structures and cause apoptosis of the cells lining the hair follicle and the hair bulb. So the experimental layout is very simple. We had skin obtained from facelift, hairy skin. The skin was divided into a positive and a control. The test solution containing hydrogen peroxide, 4%, was applied onto the skin, and then an epilator, a typical commercial epilator, was passed on it to remove the hair shafts. As a control, we had the same strength of hydrogen peroxide solution, but in an aqueous solution. Once the epilation was done, the skin was washed and incubated for 24 hours to wait for the active compound to carry out its effects on the cells. Then the skin was sectioned and stained for hematoxylin eosin for the cell morphology, tissue morphology, and cleaved caspase-3 to detect apoptosis in the cells. The results, which are highlighted here, show that in the H&E stained samples, we can clearly see there is a damage in the hair structure. This is the follicular infundibulum, with characteristic bubbles which we believe are derived from the in situ generation of oxygen, which is caused by decomposition of the hydrogen peroxide. None of this is visible in control epilated skin. If we move on to the caspase-3, the apoptosis staining, we correspondingly see an increase in apoptotic cells around the outer root sheath of the hair follicle, which is a lining that remains once the hair shaft is removed, which is very prominent and almost absent in the control epilated hair follicles, indicating that the active compound hydrogen peroxide has penetrated into the follicle, caused apoptosis, but none of this has happened in the control, meaning that the test hydrogen peroxide solution did not have a means to penetrate the follicle in the absence of the appropriately designed organic solution. Another interesting result is that the interfollicular epidermis does not seem to be affected either morphologically or biochemically by the application of the solution. And this means that the penetration of the solution within the skin was not sufficient to cause any significant damage. Also because following application, the solution was removed and the skin washed. This wash was enough to remove any solution from the interfollicular epidermis, but not the solution that had penetrated in the follicles during the hair extraction. Now, taking this one step further, this formulation was actually licensed to a company which carried out an in vivo test on some volunteers, because this formula is entirely made of cosmetically licensed ingredients, so that the hope was that the damage to the hair follicles would result in a permanent reduction of the hair growth following this treatment, and that would be visible as a reduction in hair count and hair strength. And that's exactly what was observed. Following treatment with this solution on one side versus the control treatment on the opposite side, you have almost a twofold reduction of the number of hairs coming back in the positively treated sample, thereby achieving, incidentally, permanent hair reduction, at least for a period of 16 weeks, which is about 4 months, and it's pretty significant interval. So, wrapping it up, we can actually conclude that this kind of approach is able to deliver biologically active compounds to the hair bulb and the infundibulum of the hair follicle, and can be used for a variety of molecules, hydrophilic molecules, which represent the majority of interesting drugs, including inhibitors and promoters, able to treat hair growth disorders such as hair loss and or excessive hair growth. And it is a general purpose system that can be hopefully applied to many other kinds of drugs. And with this, I conclude my presentation, thanking the people who collaborated with me, Mr. Rossella Di Santo, who helped a lot with the sections, and of course, the Mike Philpott group in the Center for Continuous Research of the Blizzard Institute, Queen Mary University. Thanks for listening.